In this video, we're going to look at DNA damage. So the first DNA damage we're going to look at is cyclobutylthymine dimer, or also called a TT dimer. So this happens when you have two thymines in a row in DNA, and you go outside in the sun. UV radiation will come and hit you, and what UV radiation can do to your DNA is that these two thymines are now connected in a cyclobutyl ring. This causes stress. This distorts the DNA base pair structure. You can't really transcript or replicate anymore. And what can also happen is that this UV damage can create free radicals, these hydroxyl free radicals, which can then go and destroy other DNA or other protein. Next type of damage we're going to look at is oxidative deamination. And when we're talking about DNA damage and mutations, we have two main classes that we want to talk about. Point mutations. A point mutation is where one base is changed into another. And insertion or deletion. New nucleotides are inserted into the sequence, or nucleotides are deleted from the sequence. So we're going to look at point mutations, and point mutations result from one of your bases being altered. So what we're looking at here is HNO2. This is a preservative that's found in prepared meats. And what HNO2 can do for cysteine is that it can knock off this NH2 deamination. When this NH2 is knocked off, it can be replaced with a C double bond O. This cytosine is now a uracil. And if this was not corrected, uracil would hydrogen bond pair with adenine. So your CG would become a UT. Luckily, DNA doesn't have uracil. It has thymine, which has a methyl group. So when we find uracil in our DNA, we actually have a specific enzyme that will take care of this uracil and replace it with a cytosine. And we'll talk about that enzyme in another PowerPoint. Here we have adenine, and HNO2 will come off, and it will become hypoxyanthine. This hypoxyacine will now pair with cytosine. So instead of an AT, now we have a C over here. And if this isn't corrected when we do DNA replication, we're going to put a G where an A used to be. So that's one way that you can have point mutations. This can also happen spontaneously in your body. This NH2 will be cut off many times a day in many cells and become uracil. So it's just not always from chemicals that you get from your diet or your environment, but this just happens naturally as well. So we're going to talk about reactive oxygen species and why they're bad. So superoxide ion, hydroxy radical, and hydrogen peroxide, these can all be created through oxidative phosphorylation, oxidative metabolism, and they can damage DNA. So one thing they can do is they can take guanine and they make it into oxo-G, or 8-oxyl-guanine. And oxoguanine can now pair with A. So it doesn't just have to pair with C. And if it pairs with A and it isn't caught, then you'll have a mutation where you used to have a G and C, now it's an AT. So that's one way reactive oxygen species can harm your DNA. You can also have alkylating agents that will generate transversions by creating O6 methylguanine. This is nitrogen mustard, ethyl nitrourea, or this N-methyl N-nitroguanidine, MNNG, and they will add methyl to your guanine residue. And if you have this methyl or your guanine, what's going to happen is that you're likely to cut your glycosidic bond that connects your sugar to your base. So this bond right here. If you lose that glycosidic bond, you could have trouble translating. You could have problems with replication. And so that's generally a thing you don't want to happen. But this is actually a somewhat common occurrence. So let's say that in each cell you have roughly 6 billion glycosidic bonds. You lose 20,000 of them roughly in a day naturally. So we do have mechanisms to correct that. And as long as you have your template strand or the strand across from this, we should be able to fill it in no problem. Now let's talk about insertions and deletions. And one way you can get an insertion or deletion is by treating your DNA or DNA coming into contact with intercalating agents. So what is intercalation? So here we have the molecule proflavin. And what proflavin can do, you can see it's a bunch of carbon rings. And between DNA rings we have base stacking. Well, this ring can go right in between this DNA and find a nice little cozy home there. And when that happens, the distance between consecutive base pairs have now roughly doubled, causing distortion of the whole DNA structure. And when you try to replicate distorted DNA, sometimes, since the distance is greater than you expect, you accidentally insert or delete one or more nucleotides in your new DNA strand. And that can have large consequences like frameshift mutation throughout your genome. And though most people agree that these mutations will happen more or less at random, though. So randomly you'll just have mutations happen to your genome. And not all mutations have to be bad, right? Because not all of your genome is encoding for something. So not all mutations have phenotypical consequences. So you could hit your non-coding GNA region, and so if you have a mutation there, that's probably no problem. The genetic code has 2 copies of chromosome, so you do have a separate copy of DNA in case something goes wrong, so you could still function. And if you don't hit protein, you're probably going to be okay moving forward. However, 80% of the cancers we see in humans may be caused by carcinogens like this molecule that do cause a mutation that do have phenotypical consequences. And just to give you an idea of the number of carcinogens out there, roughly every year there's 80,000 man-made commercial chemicals that we use, and roughly 1,000 are made every year. To really see if these chemicals are carcinogen, that they cause cancer, you usually do tests and you expose rats and mice to high levels and see if they develop cancer. This is very expensive and time consuming, as you can imagine. So even though we make 1,000, many of these substances don't go through this process. So there's actually another quicker, faster test that was developed called the Ames test. Here we use bacteria to see how cancerous these new chemicals are. So the idea of the Ames test is that you have a bacteria, Salmonella typhimurium, and these bacteria cannot make histidine. So you put them on a plate that doesn't have histidine, and you're going to see, do these bacteria spontaneously regain the ability to make histidine? If they do, they live. If they don't, they die. And so you have a culture with this mutagen for 2 days. And you also have proteins that are naturally found in the liver, because the liver can break down some of these carcinogens. So we want to account for that as best as we can, as this was a human system. And so you have your bacteria, and what you can do is that you have low levels of this carcinogen at the end, and you have a gradient, where as you move closer to the center, you have higher and higher levels of carcinogen. And what you can see is that you have more carcinogen, you have more colonies. More colonies meant you have more mutations to regain this His ability, which means it's more cancerous. So we are using bacteria for this, but it has been found that there is roughly an 80% correlation when we do these rat mice tests. So it's a good indicator if something's going to be hazardous for us. And just to show you what the Ames test has found, so tris-2,3-dibromopropanol phosphate, flame retardant in sleepwear in the 70s, gets absorbed by the skin. Through doing the Ames test, we found, oh, that's a carcinogen. Maybe we shouldn't have that in kids' clothes. And furylfermide was antibacterial additive in food in the 60s and 70s in Japan. Did the Ames test, thought that it was a carcinogen, and so we no longer use that because it can cause cancer. And that's it for this video. I will see you in the next one.